presentation. I'm going to talk about liver now. Well, we heard a lot about tumor, and I will end with the tumor also. But I'm going to talk about interferon gamma, which is a potent cytokine for the treatment of chronic liver fibrosis and tumor angiogenesis in vivo. So why I'm so interested in liver fibrosis? Because it's a growing health problem. About 1.5 million people die every year because of this. And if you see the graph here, this is the liver fibrosis, which is increasing over years, and other diseases, for example, diabetes, cancer, respiratory diseases, they are going down because there is a curative therapy available. But for liver fibrosis, there is no therapy available, and transplantation is the only option. Just to introduce a little bit about the pathogenesis of liver fibrosis starts with a primary cell, which is hepatocytes, apoptosis, which can be occurring due to viral infections, excessive alcohol intake, or obesity. This leads to apoptosis of hepatocytes, which produce lot of mediators and growth factors. And inflammatory cells, they go in the liver, they infiltrate, and this leads to the activation of otherwise quiescent stellate cells. These are called hepatic stellate cells. And these cells, they differentiate and activate, and they produce a lot of extracellular matrix within the liver, and it leads to the scar formation in the liver and lead to liver cirrhosis or hepatocellular carcinoma. So why we are so interested in interferon gamma? Well, it's a potent inflammatory cytokine. It is produced by normally all the inflammatory cells on activation. It possesses potent antifibrotic, antiviral, anti-tumor, and immunomodulatory activities. But interferon gamma failed in the clinical trials for liver cirrhosis or idiopathic pulmonary fibrosis, and the reason was lack of efficacy, and we think it's a short half-life or low doses, and severe adverse effects. So our question was, how can we improve it? First thing we thought, okay, let's try pegylation of interferon gamma. So that was the simplest thing you could do to increase the efficacy of any cytokine. What we did, we just added PEG to the cytokine interferon gamma, and we thought it will lead to enhanced solubility and stability. It will lead to prolonged circulation half-life, and it will decrease the renal clearance. So with all these factors, you would have increased accumulation of interferon gamma or pegylated interferon gamma in the liver, and you will have better therapeutic effect. What we did, we investigated the pharmacokinetics, therapeutic efficacy, adverse effects of different pegylated interferon gamma. So we pegylated with PEG 5, 10, and 20 just to see how much difference it would make to the accumulation of interferon gamma. So these are the different interferon gamma construct, and this is the dose in the blood. So this first one, let's start from bottom. This is interferon gamma alone. So you can see it is going out from the body very fast. This is pegylated interferon gamma 5. So it has an improved pharmacokinetic profile in the blood. Then pegylated interferon gamma 10 and 20. It has even improved pharmacokinetic profile. So that means we could increase the circulation time and thereby decrease the renal clearance. Then what second thing was if it has a better effect in liver fibrosis. That's what exactly we found. This is only one of the parameters which I'm showing here. It's a collagen, which is an extracellular matrix within liver. So these are, and this was done in animal model with only liver damage, so acute liver injury model. This is olive oil treated, so only a normal healthy animals. These are PBS treated, so these are fibrotic animals. So you see there is an increased extracellular matrix accumulation in these animals, and this is done in liver with different interferon gamma constructs. So only interferon gamma had a marginal effect, but interferon gamma PEG 5, 10, and 20, it has increased effect in collagen degradation, or there is less accumulation of collagen. So that showed that there is an increased therapeutic efficacy with increase in pegylation. But if there is increased accumulation of interferon gamma, you would also expect there is more side effects, and that's what we exactly saw. These are again the same animals, olive oil healthy animals, CCL4 animals, so fibrotic animals with increasing concentration of or increasing pegylation of interferon gamma. You see there is an increased systemic inflammation. So TNF alpha is basically an indicator of systemic inflammation. So we could see these animals were actually suffering. So there was more inflammation, and we could actually see that they were actually dying because of higher pegylation of interferon gamma. So this cannot be an option. Well, we could treat the animals, but there were more side effects. So what we thought, okay, let's improve it even more. So we did targeting of this interferon gamma. So what we did, normally in interferon gamma binds to its receptor, which is expressed everywhere in the body. It leads to antifibrotic effects, but also a lot of adverse effects. That's what I've shown you. But if we target this interferon gamma using a receptor which is called PDGF-beta receptor, platelet-derived growth factor receptor, which is highly expressed on hepatostellate cells when they are activated during liver fibrosis, then this interferon gamma or targeted interferon gamma will only bind to this receptor, PDGF-beta receptor, and should only show antifibrotic effect. That was the aim. So I first want to show the PDGF-beta receptor expression in the fibrotic organ or fibrotic liver in mouse and in humans. So this is normal animals, and these are fibrotic animals, and this red staining denotes PDGF-beta receptor expression. So you can see clearly there is a higher upregulation of PDGF-beta receptor in fibrotic livers. And we could see the same thing in human cirrhosis. These are normal human livers, and these are cirrhotic human livers. So we could see the target receptor is overexpressed on the stellate cells. We designed a peptide. We call it PDGF-beta receptor binding cyclate peptide. And here is the binding motif to the PDGF-beta receptor. We conjugated this peptide on interferon gamma via flexible PEG linker. So we do have PEG, but we used here only 2 kilodalton PEG chain. And this will provide the flexibility to the peptide so it can have a efficient binding to the receptor. We checked it in advanced liver fibrosis model, which is like here. So week 1, we give them CCL4 for 8 weeks. CCL4 is a carbon tetrachloride. It leads to spontaneous liver fibrosis, and we have to inject them twice a week. Control animals are only given olive oil because CCL4 is prepared in olive oil, so our control animals are only olive oil treated. For 6 weeks, we give them CCL4. That means they have established liver fibrosis, so it's a post-treatment. And then last 2 weeks, we treat the animals and we continue CCL4 treatment. So that means the fibrosis continues, and we try to treat these animals. We only used half of the dose of the interferon gamma, which is normally used for patients. So for patients, we use 5 microgram. We used only 2.5 microgram here. So, and we gave them, so these are olive oil animals. So again, I'm showing the parameter extracellular matrix accumulation, but we also did number of parameters in this paper. So collagen accumulation, so this is olive oil animals, so only healthy control. So you see there is almost no collagen expression. These are CCL4 animals, so really chronic fibrotic animals, and then you see there is a bridging fibrosis, we call it. So there is a lot of collagen accumulation. Interferon gamma had a marginal effect. Interferon gamma PEG, which is 2 kilodalton PEG, also had less effect, but our construct almost diminished collagen accumulation. This you can see on stellate cell activation here. These are the markers for stellate cell activation, and with this we could show the effect is because of the inactivation of these hepatostellate cells in the liver. But okay, we treat the animal, but what about the side effects? We studied all the reported side effects which actually human have with interferon gamma. So flu-like symptoms, so we measured fever for the animals. Neurological symptoms, we measured inflammation in the brain. Cardiovascular effects, systemic effects, so just plasma, TNF alpha and IL-6. Hematological effects, platelet counts. So these all parameters, they were either upregulated or I think platelets were downregulated, but with our targeted interferon gamma there was no change. So we could actually have better therapeutic effects without side effects. But if I go back to my slide, which is here you see that I told you that interferon gamma, when conjugated to a peptide, it will bind to PDGF beta receptor. But you would think interferon gamma can also bind to its receptor. So if we treat patients for over years, there are chances that interferon gamma bind to its receptor and start having a side effect. So we made even more improvements. We made interferon gamma peptidomimetic. This interferon gamma peptidomimetic has only the activity domain of interferon gamma. It lacks the binding domain. So this was our formulation earlier. So interferon gamma, PEG and the peptide. Then we made interferon gamma peptidomimetic, which only had the activity domain of interferon gamma with a PEG linker and the peptide. So the effect you would see would only come from the peptide mediated internalization of interferon gamma peptidomimetic. This will, by any chance, will not bind to interferon gamma receptor. So this was developed for the clinical point of view, and we also made a patent from this. Interferon gamma, then we did the same study which I told you in 8 weeks model. So we treat the animals for 6 weeks CCL4, and then in last 2 weeks we gave them mimetic interferon gamma by PPB. So this is another peptide we designed. So then I show you the similar parameter first, ECM accumulation, so collagen accumulation in these animals. So these are fibrotic animals, interferon gamma treated animals, so only marginal effect. Mimetic interferon gamma PEG, so because it doesn't have the binding motif, it will not show any effect. So therefore you don't see any effect from mimetic interferon gamma alone. And with mimetic interferon gamma targeted with bi-PPB, you see huge effect. And we could also see in the activation of stellate cells, we could see there is a good effect from mimetic interferon gamma by PPB only. And this we also saw in quantitative real-time PCR. So that was about the effects, but what about the side effects? So we did only few parameters which were already we did in previous study. MHC2 expression, so for the inflammation in the brain and inflammation in the liver, and then you see interferon gamma alone lead to MHC2 upregulation in the brain and not any other treatment, and especially not a mimetic interferon gamma PEG by PPB. So the targeted mimetic interferon gamma, and also IL-1 beta was overexpressed in liver, but there was no change by other treatments. So interferon gamma does induce effect, but also important to note that mimetic interferon gamma didn't show any effect. That means it is not active because it doesn't have the binding motif. Then we thought, okay, if it has an effect on fibrosis, maybe it also has an effect on tumor. Reason being because we know that tumor is driven by fibroblast. So we checked mimetic interferon gamma by PPB in C26 colon carcinoma model. It was a subcutaneous model we used, and we treat these animals about 6 doses after having the visible tumor seen. And then here you can see that interferon gamma didn't show effect, but in mimetic interferon gamma by PPB showed significant effect, and it reduced the tumor volume at the end of the experiment. And then we measured if what was the reason. We could see there was reduced angiogenesis in these tumors. So if you see CD31, which is angiogenesis or blood vessel marker, you could see that mimetic interferon gamma by PPB significantly reduced CD31 expression, and angiopoietin 1, which is also an angiogenic marker, you could see there was a significant reduction with mimetic gamma by PPB, and so is microvascular density was also reduced. We are doing more studies with this on tumor in different type of tumors. So in conclusion, I showed you that pegylation of interferon gamma drastically improved the therapeutic efficacy, but also led to systemic inflammation. Stellate cell specific targeting of interferon gamma to fibrotic liver increased its therapeutic efficacy and eliminated interferon gamma related adverse effects. Mimetic interferon gamma coupled to the peptide targeting peptide induced potent antifibrotic effects in liver fibrosis models, and their adverse effects were gone. And targeting interferon gamma, mimetic interferon gamma could also reduce tumor angiogenesis in colon carcinoma model, which provides broad opportunity to use this as an anti-cancer therapeutic. So finally, I want to acknowledge some people who did this work, including myself. And thank you for your attention. If you have question, I would be happy to answer.